Okay, now that everyone's here, we can start. So after a short break, I want to thank you all on behalf of the Student Society for Molecular Biology and the Serbian Society of Molecular Biology for attending today's lecture titled Functions and Modes of Long Non-Coding RNAs in Mammalian Cells. Today with us, we have Professor Igor Olitsky. Professor Igor went to the Tel Aviv University, where he got his bachelor's degree and a PhD in computer science and life sciences. After that, he was a postdoctoral associate at the Whitehead Institute for Biomedical Research that is affiliated with MIT. Currently, he is an associate professor at the Weizmann Institute of Science, where he runs his lab. The main focus of his lab are diverse mechanisms in which long non-coding RNAs act, and their main goal is understanding their functionality and the ways it manifests itself in their sequences. They also have other interests, such as other areas of gene regulation, including nuclear export and localization of long non-coding RNAs, chromatin biology, and RNA decay. To study these questions, they use various systems, including human cell lines and primary cells, mouse embryonic stem cells, and mouse models. His lab is known for combining both tools from experimental and computational biology. If you have any questions, I will ask you to send them via chat to everyone, or to ask them at the end of the lecture. You can turn on your camera, or you can just turn on your mic. Professor Igor, thank you for being here with us today. The floor is all yours.
Thank you. So thank you all for connecting, and thanks, Todor, for the invitation. So I want to talk a bit about long non-coding RNAs and why these molecules are interesting, why we are excited about them, why other people are excited, and also why these are relatively challenging genes to work with. So I'll give both some background and focus in a bit more detail on two different studies that we have done on lincRNAs, one more from the computational side, one more from the experimental side. So feel free to either type in questions in the chat or even stop me, and I'll be happy to answer questions. I'll also try to stop in the middle a couple of times, and I'll try not to speak too fast, which is usually a problem that I have. So one reason that we're interested in understanding these long non-coding RNAs is that if we look at the human genome, only about 1% of the genome, so of the 3 billion bases in the genome, only about 1%, about 30 million bases, are dedicated to encoding information about proteins. So if we look at the exons of the genes that are encoding proteins, take all of these exons together, that's only 1% of the genome. A bigger portion of the genome is found in so-called introns. So introns are pieces connecting exons together. That's about 40% of our genome. And arguably their main function is to be part of the process of biogenesis of mRNAs, which go on to produce proteins, even though introns will also sometimes have other functions. So, but the majority of the genome is then in this so-called intergenic space. But if we look at mutations in the human population that are associated with different diseases, with, for example, the probability that we will develop diabetes in our lifetime or develop different neurological diseases and so on, about 45% of these mutations are found in these intergenic regions. So for many of these mutations, for the vast majority of them, we don't really know what is the actual functional change that is responsible for the disease or for a specific human trait, because there is a lot of linkage between different positions. But for these about 45% of the cases, we know that wherever the change is, that change doesn't change the sequence of any protein. So these changes are all regulatory, and presumably many of them are acting on the DNA level. So that means that they're going to affect enhancers or promoters and so on. But we also know from studies in the past couple of decades that a large proportion of these regions are transcribed into RNA molecules. So that is that they're producing RNA, but this RNA does not go on to produce functional proteins. So there's a lot of non-coding transcription. We can wonder whether it's functionally important or not. I'll be talking about that quite a lot, but we're certain that major parts of our genome are transcribed into RNA molecules that do not produce functional proteins. So there is a lot of real estate that is producing RNA. And then we can ask what kind of RNAs are being made. So one type of RNAs that are being made are the so-called small encoding RNAs. These include microRNAs that you probably heard about, and these have been discovered in the beginning of the century and have been extensively studied and are now really very well understood. So we know exactly how they're processed and what they can do. They typically begin as long RNAs, get processed into small RNAs of about 20 bases. These can go on, pair with specific transcripts in the cell, and typically downregulate their expression. These also have a lot of therapeutic potential and sort of harnessed to silence genes, including in recently approved FDA drugs. So this pathway is relatively well understood, and there are several additional types of small RNAs that are being made. But as their name implies, they only explain a very small portion of the transcription observed in the genome. There are longer RNAs of intermediate size. These are typically around 100 bases, and these are some of the best studied RNA molecules. So tRNAs, snRNAs, snRNAs, and so on carry out key functions in processes such as translation and splicing, very well understood. And as I'll describe in my next slides, are also quite different than the group that we are really interested in, which are the long encoding RNAs. So here, technically, ribosomal RNAs fall into this category, and these are very well understood. 2/3 of the ribosome is RNA. This is a very detailed and very important complex formed around RNA. But the vast majority of long RNAs, as I hope that I'll be able to convey in this lecture, they don't really look anything like ribosomal RNA. Their features are really different. So the vast majority of these RNAs that are transcribed have only been described relatively recently, and for the vast majority of them, we don't really know what they're doing, if anything. So the ones that we are focusing on are the so-called lincRNAs. They're technically defined as being at least 200 bases long. Typically, they're longer. Typically, they're about 1,000 bases long, and some of them can be as long as 100,000 bases. So the first discovered examples were already in the 80s, and there was one lincRNA, and I'll go back to it, discovered in the 90s called Xist. But for a long time, these were very few examples. So maybe 4 or 5 different lincRNAs were described in the 90s. And then about 15 years ago, when people began developing approaches which allowed in an unbiased way to capture RNA in cells and sequence it, it was discovered that actually there are many such loci in the genome which are being transcribed. In many cases, they're not transcribed very highly, so they're only producing few RNA molecules per cell, and so it was difficult to see them before these different approaches were discovered, which allow a more thorough interrogation of what kind of RNAs are produced in cells. But once these studies sort of began ramping up, it was realized that actually there are many different places in the genome which are producing lincRNAs. So at least 20,000 different human lincRNA genes compared to about 18,000 genes that are producing protein coding genes. And as more and more studies are done on different tissues and different cell types are profiled, the number of these lincRNAs is growing, whereas for protein coding genes, more or less, what has been discovered has been discovered. We don't typically find many new protein coding genes. Again, typical length, about 1,000 bases, and typically they have 2 or 3 exons. So this is also in contrast to protein coding genes, which typically are going to be a bit longer. They're going to have 7 or 8 exons on average. And as I already mentioned, one reason that they haven't been discovered before is that most of them are not really abundant. So a typical mRNA is going to be found at about 10 to 5 copies per cell on average. A typical lincRNA is going to be about an order of magnitude lower, so about 1 copy per cell, sometimes less than 1 copy per cell on average. And most lincRNAs are much more tissue-specific than mRNAs. So in terms of rules of thumb, we have, as I mentioned, about 18,000 different protein coding genes. In any tissue or cell type, they're going to be expressed about half of them. So about 10,000 different proteins are expressed in any cell type. So if you look at a brain cell or a blood cell or a skin cell, about 10,000 proteins, about half of the proteins encoded in the genome. If you look at lincRNAs, there are again more than 20,000 of them, but any cell type is going to express only about 1,000. So maybe around 5% of what it can, which is a much lower number, as you can appreciate. So expression is much more tissue specific. Of course, there are exceptions. There are some lincRNAs that are going to be expressed highly. Some of them are going to be expressed in every cell type, but the typical lincRNA is much more tissue specific than the typical mRNA. In terms of their stability, there is also a lot of variability. So there are going to be some that are going to be as stable as mRNAs, so stick around in cells for hours, but others, the majority of them are going to be somewhat less stable. They're going to stick around for a few minutes. And the same goes for how efficiently they undergo splicing. So a typical lincRNA undergoes splicing similarly to an mRNA, but typically the efficiency of that splicing is not as high. So if we now can compare sort of several features that are really strikingly different between the classical kind of intermediate size RNAs, so these are tRNAs, noncRNAs, snRNAs, or ribosomal RNAs that carry out known key functions in cells, and this large collection of all these new RNAs, lincRNAs. Some of them are antisense to other genes, and generally speaking, circular RNAs that I won't talk about, they also resemble all these different features. So for many of the classical noncoding RNAs, they're produced by polymerases which have been evolved to specifically produce noncoding RNAs. So Pol3 is transcribing tRNAs, Pol1 is transcribing ribosomal RNA. These are really dedicated for production of these RNA molecules, whereas lincRNAs are typically produced by RNA polymerase II, which is the polymerase that is producing mRNAs. So that's why most of them are spliced, because this is the polymerase that is typically producing spliced products. And this is also one reason they're not so abundant. Pol2 usually does not produce a large number of RNA molecules in contrast to Pol1 and Pol2. Again, this key difference in abundance I already mentioned. So these are relatively lower abundance than mRNAs, whereas classical encoding RNAs are typically very abundant. So microRNAs, when they are functioning in cells, are typically found in thousands of copies per cell, tens of thousands of copies. A lincRNA, often 1 copy per cell. In these classical encoding RNAs, they're clearly defined families. So tRNAs, noncRNAs, snRNAs, they have specific defining features, specific structure they fold to. So even when their sequences are not similar, there is something really clearly shared between them. And this is typically the key to their function. So a tRNA is going to fold in this cloverleaf structure, and then the amino acyl transferases, the proteins that are loading the tRNA with amino acids, this is what they're recognizing. So they're recognizing the specific fold of the tRNA. In lincRNAs, they're certainly folding into different structures and so on, but so far we as a field haven't really been very successful in sort of identifying these families and separating them, lincRNAs into these families. In classical encoding RNA, structure is really important, again, as with the tRNA example. In lincRNAs, often it's less clear. So they're going to fold into specific structures in cells. Even if you take a random RNA sequence, it's going to fold into a specific structure. But how important that structure is remains questionable in most cases. So there are some examples where it's clear, but in many cases, it's not clear. Intermediate the RNAs are typically highly conserved. Some of them are conserved between bacteria and mammals. Again, tRNAs, snRNAs, ribosomal RNA, and so on. And lincRNAs are often evolving much more rapidly. And as we'll see, there are many human lincRNAs that are only found in human or found in human and in primates, but not in further species. And in fact, today we don't have any lincRNA that we really think is conserved between vertebrates and invertebrates. So for example, between human and flies. Whereas there are many proteins that are conserved between human and flies, and also microRNAs and other non-coding RNAs that are conserved. So lincRNAs are evolving more rapidly. But despite these characteristics, which might be suggest that they're not so functionally important, so low expression levels and high evolution rate, there are now many processes where lincRNAs have been implicated. So every year there are more and more papers showing that lincRNAs, when you knock them down, in many cases this is going to be in some cell line, there is an effect, something happens, suggesting that they are functionally important. And if we look at where they're functionally important or where they're active in general, there is evidence that many lincRNAs relatively are active and expressed in the brain, and specifically in different types of neurons in neuronal cells and so on. So this is a heat map showing different lincRNA genes and different tissues. And we can see here that there are many lincRNAs expressed in the testis. In general, sperm and different cells during development of sperm are expressing typically almost every gene that is encoded in the genome. But except for testis, the brain has a large repertoire of lincRNAs that are expressed. And if we zoom in into different types within the brain, that is the case as well. Okay, so when we're working with lincRNAs, again, some of it we're doing from more sort of bioinformatics, computational perspective, and other genes we're looking at them experimentally. So one question that you might be or maybe should be asking yourself is, how do we really know that they are really non-coding, right? So if we found now a gene and this gene is transcribed into RNA molecules, how do we know this is really a non-coding RNA? So typically computationally, we can use a number of different filters. So first of all, we can ask, does this sequence have a long open reading frame? So open reading frame is a part of the RNA sequence that begins with the start codon and ends with the stop codon. And in most proteins, they're quite long. So most proteins are going to be typically at least 200 amino acids long. And this is much, much longer than what we would expect by chance. So by chance, because there are 3 stop codons and 60 codons in total, by chance, every 20 codons, there should be a stop codon. But if we now have a sequence that has an open reading frame of 200 amino acids, that's 10 times longer than what we would expect by chance, more or less. So that's one feature that is typically associated with proteins. But it's not enough because they are small proteins. So just if a sequence only has a short open reading frame, doesn't mean it doesn't make a protein. So we can also look at other features such as what is the composition of the codons in the sequence. And the feature that's probably most useful is we can look at how this sequence is evolving. So we can take the sequences of the same region from different species, compare them, and then ask, does the pattern of changes in this sequence resemble what we would expect to see in the protein? So the protein we would expect to see typically 2 bases that are conserved, and then the 3rd base that is changing more rapidly. And if there are insertions or deletions, we would expect them to be divisible by 3. So these features can be used to detect even relatively short regions that are making proteins. Also, proteins are typically similar to each other, so we can look at similarity between proteins or proteins encoding specific domain as evidence that this is a protein. And typically, things that survive, all sequences that survive all of these filters, they are what is referred to as lincRNAs. So it's a challenge on one hand, because this is all definition by what they're not, right? So in contrast to some other genes, we cannot say that there is some feature that once that feature is found, this is lincRNA. We can only say if it doesn't have this and doesn't have this and doesn't have this and not this and not this, then it's lincRNA. So, but still this is operationally in the vast majority of cases good enough, and the sequences that survive these filters, they typically do not encode any thing that resembles a functional protein. So we can be quite certain that at least if we think about long and reasonably conserved proteins, typically also short proteins, it's unlikely that we're missing too many of them. And if we look at complementary experimental data, such as mass spectrometry, it's typically that only very few of sort of things that are annotated as lincRNAs appear to be producing any functional proteins. So we can be quite confident that these are indeed thousands of non-coding RNAs that are being produced. And what my lab and others are trying to ask is, what kind of activities can these RNAs carry out in cells, and how these activities are encoded in their sequences and maybe their structures. And from the biomedical point of view, this is an important question, because eventually one thing we would like to do is we would like to interpret genome sequences, right? So today it's quite cheap and quite accessible to sequence human genomes, and in many countries quite soon, every baby that's going to be born, there's going to be an option to sequence his or her genome. But and if that individual has a certain condition or a certain syndrome, exome sequencing is now almost routinely used in many countries to identify which mutation is causing that particular condition. And exome sequencing is only looking at this 1% of the genome that is encoding for proteins. And nevertheless, in about a third of the cases, exome sequencing is working. So that means that the mutation that is causing the condition is found. And the third is actually quite a lot, but the assumption is that these other 2/3, where an individual has a certain condition, but exome sequencing doesn't seem to identify any mutation in the protein coding gene, the assumption is that many of these cases, the mutation or the deletion or insertion of a region are in this intergenic space. And there, even though the ability to sequence whole genomes is there, we're still completely lacking sort of a capability to go and say, this mutation is likely functionally important because it's taking this lincRNA gene and it's affecting a region or a base in that lincRNA gene, which is doing something functionally important, right? So exome sequencing in protein coding regions works because we understand how proteins are being made. We know what kind of mutations are nonsense mutations or missense mutations. We can know the functions of many proteins. We know what kind of mutations are really going to have a strong effect on the protein. In the intergenic region, we typically don't have that ability. And that is a major challenge for the field. And the other thing is we'll show that there is a lot of motivation for identifying which lincRNAs are functionally important, because there are ways today with CRISPR or with antisense oligonucleotides to manipulate their expression, right? So if we have a lincRNA that is doing something functionally important, maybe we can reduce its expression or increase its expression, and this can have a benefit as a potential drug. But for that, we need to understand what kind of activities are they're carrying out, or where are functionally important regions in them. So my lab has been sort of really combining sort of everything all the way from computational biology into establishing and analyzing specific mouse models to try and address this question. So one challenge in the field, and sort of again, I keep going back and forth between the promise and the challenges. So one challenge is that lincRNA loci can act at multiple different levels. So when we think about RNA biology, traditionally we like to think about sort of the functions of the mature RNA. So an RNA is being made, doesn't matter where it was made, it's going to go somewhere and carry out a certain function. But for lincRNAs, we know that in many cases, what's going to be functionally important is not really the RNA product, but maybe just the act of transcription of that RNA. Or maybe the RNA is going to be functionally important, but it's only going to act in the context of the region of the genome where it was produced. And this is sort of more challenging to study, and it's also quite challenging to determine at what level this is acting. And part of the challenge is that if it's just the transcription that is important, then the sequence of the RNA is not really important. The RNA itself is not really important. If it's on the other hand, if it's transacting RNA, then only the RNA is important. Then it doesn't matter where it was produced. And for these nascent RNA functions, it's somewhere in the middle, right? So the RNA sequence might or might not be important. So how can it be? How can just the act of transcription be important? So we know that RNA polymerase, as it is producing RNA, can physically interfere with other factors. So for example, so we can sort of imagine a road, and RNA polymerase is this truck which is sort of driving on that road. So as it is driving on that road, it can dislocate different factors. It can also affect the underlying chromatin. So it can go and change, deposit different modifications. In some of these modifications, for example, if there is a methylation, this is a chemical modification on lysine number 36 in a particular histone protein, this modification is known to be found within regions, long genes that are being produced, and it is inhibiting transcription initiation. So if we have a lincRNA which is transcribed, it is possible. So within the region which is transcribed, the chromatin is going to be changed, and now it's going to sort of prevent different promoters in the same region from being active. And indeed, with this has been shown to be the case sometimes. So there can be a negative effect on other promoters, but transcription can also help open chromatin. So we can also imagine cases where there's going to be a positive effect. So this is one example for this. This is a case where of so-called imprinted regions. So regions in the human genome where, so most genes that we have they've encoded on 2 chromosomes, and both of these chromosomes are transcribed, and both of them are producing RNAs, right? So we have one chromosome which we inherited from our mother, and another which we inherited from our father, and both of them are active. But there are cases in the genome where a so-called imprinted loci, where only the maternal or only the paternal chromosome are transcriptionally active. So this is one such region. And here on the paternal chromosome, the gene, which is called IGF2R, is being repressed. And it is repressed by an RNA, by a long RNA called AIR. This one is very long, it's about 90,000 bases long. And it was shown that the mechanism for which this is acting is that transcription of AIR is sort of physically changing the chromatin in this region and then preventing this promoter from being active. So on the paternal chromosome, we're only going to have this lincRNA being transcribed and not the protein coding gene, IGF2 receptor. Whereas on the maternal one, the lincRNA is silenced. And so this gene is expressed. And right. So what kind? So this is for the active transcription itself. As I mentioned, the second layer can be that it's not the active transcription that's important; it's the RNA, but it's the RNA that is acting within the context of the region where it was transcribed. So a nascent RNA that has just been made, is carrying out the function. So the kind of activities that have been shown for these nascent RNAs, for example, is that they can recruit specific factors and then kind of take them for a ride across the transcribed locus. So recruit specific factors, and now this is increasing the local concentration of these proteins, and now these proteins are doing something else in the vicinity of where they're found. And in principle, it can also recruit. So again, this is kind of similarly recruit different factors to these loci. It can also have another negative effect. Maybe it is repelling different factors away from chromatin. And sort of one particularly challenging, particularly attractive model is that it can also be a target for some combinatorial recognition of proteins. So one very well-studied example of this is a repressive complex called polycom. So it is a complex that is typically repressing chromatin or depositing marks that are repressive. So there is a model for its function that suggests that once it sees that there is RNA being produced in a place where there are these repressive chromatin marks, it's going to enforce, sort of deposit additional repressive marks, right? So it's recognizing this combination of RNA being produced in a region where there shouldn't be transcription. And once it is sensing that, it is going to deposit additional silencing marks. So again, we can imagine various sort of complex combinatorics that can happen. And again, in some cases, they're going to happen specifically at the nascent RNA level. In some cases, because the RNA which is produced is just going to be stable, so it's not going to really stick around much longer after it was produced. And in other cases, because it's actively being retained at its site of transcription. So the classical example of such a sort of nascent cis-acting RNA is the Xist lincRNA, which was one of the first lincRNAs to be discovered about 30 years ago. And it is very important because it's essential for life in mammals. And it is produced specifically in female cells. So one of the challenges that mammals have to deal with, many other animals have to deal with as well, is that there are many genes that are found on the X chromosome. And on the other hand, cells need to sort of function in many cases in a similar way, whether they are male cells, so they only have one copy of the X chromosome, or female cells which have two copies of the X chromosome. So for many functions, so you can imagine that you have many complexes in cells. You can think of them like as a car, and this car is produced from many parts. So the different proteins are different parts of that car. And so you have two different ecosystems. You have a male cell where some of these parts are only produced by, let's say, one factory. And then you have female cells that now have two factories producing these parts. So if you eventually want to build complete cars, that poses a challenge. And in mammalian cells, the way that this challenge is being solved is that one of the two X chromosomes very early in development is randomly selected, and that chromosome is silenced. So basically, you're shutting down half of the factories. And the molecule that is really kind of solely responsible for starting this process is a lincRNA called Xist. And sort of one of the manifestations of this process of sort of exon activation in general are these nice cats that have three different sort of differential colorization because the gene for the fur color is encoded on the X chromosome, and because this X inactivation is kind of a random process, so in some of the cells, one X chromosome is going to be silenced and some the other. This is what leads to this nice, I think it's called calico pattern of fur color. But as I mentioned, this process is really essential for life. So without Xist, without silencing one of the X chromosomes, an embryo cannot develop beyond a certain stage. And so this process, again, is initiated by transcription of the Xist RNA. So initially, there is this random selection process, kind of like a coin that is being tossed. One of the X chromosomes begin transcribing this Xist lincRNA. Initially, it is found near where it is produced, but then it is spreading and can be microscopically seen to cover one of the two X chromosomes. And then later on, additional factors are being recruited. They're changing the chromatin. Eventually, DNA methylation happens as well, and one chromosome sort of becomes completely silenced. But the process begins by a lincRNA, and this lincRNA is only going to act on the chromosome from which it is transcribed. So it's so-called cis-acting RNA. So this is one kind of extreme example of a very strong effect that lincRNA can have, but also a very local effect. It's only going to act on this chromosome. It's not going to diffuse and repress the other chromosome at all. And this was beautifully shown maybe 15 years ago already, that if you take this gene and now put it on another chromosome, put it on chromosome 14 in mouse, then chromosome 14 is going to be silenced. So this region is really sufficient for silencing. It is also a very complex process and region, so there's still a lot of active research into trying to understand how this actually works. So if we think about different mechanisms of actions that has been proposed for lincRNAs, so these are transcripts that are being produced from regions that typically don't overlap other protein coding genes. So one mode of action would be this kind of co-transcriptional cis-acting regulation. So the production of a lincRNA is going to have a negative or a positive effect on the expression of genes nearby. Again, Xist is an extreme example where it's going to silence all the other genes, or almost all the other genes that are transcribed from the same chromosome. Another mode of action potentially can be that if this lincRNA can go and somehow find specific other regions, it can also recruit different positive or negatively acting proteins to these other regions. So it can act as kind of a guide. Here, the challenge is how these RNAs are actually recognizing distant loci. We don't really understand how this is working. There are going to be some cases where this RNA is going to act as a scaffold, and then different proteins are going to meet together and sort of assemble a different specific body based on this RNA. This is typically going to happen when this RNA is relatively abundant. So there are not many cases like this, but some of the best studied lincRNAs, this is what they do. This has mostly been described in the nucleus, but there are examples now of this in the cytoplasm as well. In some also rare cases where the lincRNA expression is particularly high, it can act as kind of a decoy. So it can have a large number of copies that a specific factor might bind to them. And this binding is going to prevent these factors from going and doing other things in the cell. So this way, this lincRNA can act as a decoy or a sponge. Again, this would require quite high expression levels. So the thought is that this is not very common, but there are now many examples in the literature of this as well. And one of the things that RNA in principle can do best is hybridize with other RNAs, right? So it can also both fold, but it can also hybridize with other RNAs. So there are many reasons to think that this is also going to be important in many cases. These RNA-RNA interactions with other RNAs can affect the stability of other RNAs or their translation levels, and there are now examples of this as well. And of course, there can be other functions which haven't been described yet. But this is the main kind of repertoire of the different activities that lincRNAs can have. Again, some of them are going to be in the nucleus and others are going to be in the cytoplasm. So some of the key open questions in the field is, one is kind of, I alluded to, so how many of these are actually functional, right? So we have at least 20,000 different lincRNA genes in the human genome, but how many of these are just transcribed because if you're producing a little bit of RNA just in the liver, maybe doesn't really have a very strong effect. And we think that definitely many of them are likely not functional. And from the ones that are functional, in how many of them it's really the RNA product that's important. So as I mentioned, the process of transcription itself can have various effects. So we would like to know sort of where is really the function of the RNA. And then these RNAs, once they're functional, so how many different mechanisms are they employing, and what are these mechanisms, obviously? And are these mechanisms relying on the RNA binding a specific protein or binding a specific piece of DNA, or maybe both? And then, of course, how it is recognizing a specific protein or a specific RNA molecule and so on. And then eventually, sort of whatever interactions are entailed in this function, how these functions encoded in the sequence or structure, right? So are there specific regions of the sequence that are important? Is the way that the RNA folding important? And eventually, again, if we go back to human genetics, if we now go and change the sequence, what functional effect is it going to have? So these are all sort of very, we think, very interesting and challenging questions. And importantly, for proteins, we can answer now many of them. And for microRNAs also, we can answer many of these questions. But so far, we're not there yet for lincRNAs. So this is what's motivating us a lot. So I'm actually going to talk about two studies. It says here 3, but I'm actually going to talk about these 2 because I don't want to take too much of your time. But before I go on, if there are any questions, this would be a good time to so if there are any sort of global questions about lincRNAs before I dive into these 2 stories. I'm going to dive into these 2 stories. So the first study that I'm going to talk about in more detail is our attempts to understand lincRNA evolution in general. As I mentioned, it was noted early on, so from some of the first studies that identified lincRNAs, that lincRNAs tend to change in evolution more rapidly than protein coding genes. So in order to understand better what kind of changes we see in lincRNA genes, one of the first things that we did when I started my lab at the Weizmann Institute was to take a lot of publicly available data. There was also a bit of data that we generated ourselves, but most of it was publicly available from a variety of different species separated from humans by different evolutionary distances. So most of these are vertebrates, which diverged about 400 million years ago, but we also had kind of as an outgroup sea urchin, which diverged about 500 million years ago, and sea urchins are not vertebrates. In each of these species, we took RNA-seq data from at least 10 different tissues. So obviously, this is all from different studies, so they're not always directly comparable, and there are many different caveats here, but this was good enough for our purposes. And in each of them, we went and built a computational pipeline to identify lincRNA genes. So basically, we can take the reads from RNA-seq, that are typically somewhat short, use them to assemble these transcript structures, and then once we've solved these puzzles, we have a collection of transcripts. We can take these transcripts through the different filters that I mentioned earlier, identify the ones that are lincRNAs, and then first look at their numbers. So one thing we just saw is that there is a lot of variability in the number of lincRNAs encoded in the different genomes. So whereas if we look at protein coding genes, as I mentioned, in human, we have about 18,000 protein coding genes. And if we look at other species, the numbers are quite similar. So birds have a bit less, and then fish that have undergone some duplications in their genomes, they have a bit more, but this number is quite stable. In contrast, if we look at lincRNAs, numbers are quite different. So mammalian species that have larger genomes with many transposable elements, they typically have more lincRNA genes than other genomes. And whereas in some of the fish genomes, which are much smaller, so some of these are 10 times smaller than human genome, they're going to have a smaller number of lincRNA genes. But when we began comparing these lincRNA genes to each other, one sort of striking thing we saw is that in each species, the majority of lincRNAs were relatively new. So the majority of human lincRNAs were not similar in any way, in any sequence way, to lincRNAs that are outside of primates. And the majority of the dog ones, they were not similar to anything else. The majority of the chicken ones, they were not similar to anything else. And this is in contrast to protein coding genes, which in the vast majority of cases are shared between closely related species And even if you compare a human and zebrafish, for example, about 70% of the proteins are conserved. So we see that during evolution, these lincRNA genes are sort of born and die in our genomes all the time. So there is a constant turnover. But against this backdrop of a lot of turnover, we could see that there were some lincRNAs that are conserved. So the numbers here next to each node are indicating how many different lincRNA genes we could trace to each of these nodes in the evolutionary tree. So there are about 1,000 lincRNAs that are conserved between different mammals. Again, about 5% of all lincRNAs, and about 300 which could be traced to the split between to the root of amniotes. So this is shared between birds, reptiles, and mammals, and about 100 that are shared between all vertebrates. But then there were no lincRNAs which were conserved outside of vertebrates. So if you try to compare even sort of fish with sea urchin, or any of these species with sea urchin, but also with more distant species, such as different chordates or flies or nematodes, each of these other species, they have lincRNAs. So they have many lincRNAs sometimes, but we cannot so far find any similarity and say, okay, this is the same lincRNA and it has been there for more than 500 million years. And it could be that this does happen, but again, but then the similarity is lost at some point, so we can no longer really detect these cases. So again, the conclusion is that there is a rapid evolution of the repertoires of lincRNAs in animal genomes, but against this backdrop, there are some lincRNAs that are better conserved than others. What's interesting further is that even for cases where the lincRNA is conserved to a certain level, in many cases, its sequence is quite variable between species. So this is one of our favorite lincRNAs. It's called PVT1, and is transcribed in the genome from a region which is next to MYC. So MYC is a protein coding gene. It's a very famous and very important oncogene. So this whole region is typically amplified in different cancers. So MYC is relatively very well studied. And MYC, like many other transcription factors, is transcribed from a region in the human genome which doesn't have any other genes next to it. So this is MYC. This is about 400,000 bases. But even if we go way further, if we look at about a million bases upstream and downstream of MYC, we still are not going to find any other genes. So it's a very big gene desert, and MYC is sitting in the middle. But if we look downstream of MYC in all of these different species, we always see a long lincRNA which is being produced everywhere. But if we start comparing the sequences of these, we can see that the human PVT1, so this is what the lincRNA is called. It was also actually discovered in the 80s, is similar to the one in mouse, in rabbit, in dog, and in possum, but not further. If we compare fish, different fish are similar to each other, but they're not similar to mammals. And again, this is typical. But even if we compare within mammals, so so this axon is similar, this axon is conserved, this one is conserved sometimes, but then many other axons, many other parts of the mature sequence of PVT1, they're species specific. So they're only found in human, or only in mouse, or only in dog, and so on. And in many cases, this is because they're derived from transposable elements. So they're derived from these selfish DNA elements that have been sort of independently expanding in different genomes. But this suggests that eventually the sequence of the human PVT1 is quite different than the sequence of PVT1 in mouse, rabbit, dog, and so on. And this, in proteins, this almost never happens, right? So if we have the same protein in different species, sometimes it's going to have small mutations or small differences between them, but it's very unlikely that sort of whole half of the sequence is different. But this is very common in lincRNA genes. And to us, one conclusion that this suggests is that it's unlikely that many lincRNAs are kind of like this ribosomal RNA. So like this machine or scaffold that really needs to fold in a very particular way such that other proteins can join. And then this is the basis of some complex molecular machine. Because if this was the case, we wouldn't expect their sequence to change so dramatically. So big chunks of the sequence lost or gained during evolution, especially from transposable elements. And again, not all lincRNAs are like this, but many of them are. So transposon amendments heavily rewire these lincRNA loci. And even a lincRNA which is found sometimes in multiple genomes, in many cases, its sequence is going to be drastically different between different species. But sort of conversely to this, we and others have shown before that even if you have relatively small pieces of sequence that are shared between, for example, mammals and fish, that can sometimes be enough to maintain a conserved function. So if it is still the same RNA, it is likely that it is still carrying out the same function, even functioning in a similar way, even if its sequence is drastically changing. And one explanation for this could be that if this RNA, and this is what we think is the case for PVD1, so if the RNA has a function, a sort of a cis-acting function, if it really acts as a nascent RNA, in this case to regulate the expression of MYC, maybe it doesn't really matter what much of its sequence is, right? So you just need some functional bits because they're really going to be only active in this particular context. Again, maybe just from the act of transcription. And in other cases, we think that maybe there are specific regions that are functionally important, but they really don't care that much about what are the sequences around them. So they're going to act no matter what is their context. And I'll show some examples of that. And so the other thing that it suggests, if we look at PVD1, is that sometimes there are cases where it's really the same gene that has been conserved in multiple species, but just the sequence similarity at some point is becoming so weak that we can no longer detect it. So then we can ask, does this really happen? So do we really have many cases where there are different lincRNAs that don't have any sequence similarity between them, but are found in sort of corresponding regions in different genomes? And we can also computationally look for that. So we can count how many cases we have of a lincRNA, for example, in human, and another lincRNA, let's say in chicken, which are found in the same relative orientation to a conserved protein coding gene. So we can count how many of these cases happen in the genome and compare it to how many cases we expect by chance, right? So if we now just take two lincRNAs and sort of randomly redistribute them throughout the genome, how many such cases we're going to get. And we can see that, and then we can ask for the ones that we found to be synthetic, how many of them actually have sequence similarity. So we can see that for mammals, the number of these synthetic cases that we find over what we expect by chance is more or less similar to the number of the ones that have sequence conservation, suggesting that in mammals, typically, if two lincRNAs are really conserved, so they are found in the same position, usually they're also going to have sequence similarity between them. But as we compare human to more distant species, we're seeing that is no longer the case. So there's typically more lincRNAs found in corresponding positions, and very few of them have some sequence similarity, suggesting that kind of similar to PVT1 and similar to this example downstream of another transcription factor called FOXA2 here, there are many cases where the lincRNA has been there for 300, 400, maybe 500 million years, but when we're now comparing sequences that are far away from each other, we now don't have any ability to detect any sequence similarity, just eroded to a point where we're not able to detect it anymore, but the gene is more deeply conserved. So these are the numbers we can see here. They're not very large numbers, but they are lincRNAs that are functionally conserved. But because some of the such synthetic cases are expected by chance, it is more challenging to decide which of them are functionally important. Okay. So to summarize what we can learn from this kind of comparative, more computational analysis. So we can learn that we can sort of have some initial classification of lincRNA genes into different classes. So one class are going to be cases where the lincRNA has multiple conserved regions, for example, conserved splicing pattern. These are relatively rare, but if we think about functions as a particular molecular machines, these are the most likely candidates to carry out such functions. And indeed, if we look at these RNAs, they tend to be also more highly expressed. They tend to be more broadly expressed and have less overlap with transposable elements. So this is a relatively small minority, but these are the RNAs that we think are most likely to really function as an RNA molecule. Then there are more cases where just a single region of the lincRNA, or just a few regions are conserved, but then the rest of the sequence is changing more extensively. In many cases, transposable elements, add new exons, or exons are being lost. And these RNAs in more cases are more tissue specific and less highly expressed. And we think that these are more likely to maybe be cases where just the act of transcription is important, or just the nascent RNA is important. And because the RNA is really quite different in different species, whereas it is presumably still carrying out the same function. And then there is a larger class where just the position seems to be conserved. The RNA sequence seems to be evolving quite randomly. And so here again, less likely that RNA is a function important. And indeed, these cases tend to be more lowly expressed, more found only in few tissues and so on. And lastly, there are lincRNAs that are not conserved. So these can still be functionally important. Maybe they're doing something specifically in humans, right? We know humans are different from other species. But because there are more of them, and because their expression levels are again lower and more tissue specific, it is more challenging among these to decide which ones are or are not functioning. And if we now sort of go again to the positive side, so there are about 1,000 lincRNAs in this group. And these are the ones we think are most likely to be both functionally important and have some RNA-based function. And this is still a very large number. So we've been sort of going through them, sometimes one by one, sometimes by conducting different screens and trying to understand better their functionality. Any questions before I go into the second story? Okay, so I'll go into this. Okay, so this was more computational, and now it's just a more experimental story. So again, if you go back, part of our interest was mostly trying to understand these conserved lincRNA genes. Again, there are not that many of them. But another question we can ask is, where are these lincRNA genes found in genomes? So which genes do they tend to be found next to? And here, one thing that we saw when we compared lincRNAs from zebrafish and mouse and human was that there are certain gene categories which tend to be neighbors of lincRNA genes. And these are particularly genes which are involved in the process of transcription. So some are transcription factors like MYC that I showed earlier, but some of them are more general transcription related proteins, chromatin modifiers and so on. In many cases, in proximity to the genes that are encoding them in the genome, there are lincRNA genes. So this has motivated us quite some time ago to become interested in this particular lincRNA, which is transcribed from a region which is in close proximity to the promoter of a gene called CAG2. Right? So in this case, this is CAG2. It's a very long gene, so I'm not showing all of it. I'm showing just the very beginning. This is the beginning of CAG2. And upstream of it and on the same strand, there is a lincRNA, which we named Chaser, which is being produced. This is the beginning of Chaser. This is the beginning of CAG2. And again, they're transcribed in the same direction. And if we look at Chaser, it has 5 exons in most cases in human. And if we go and look at mouse and pig and lizard and frog and zebrafish, it is quite similar in these other species as well, right? So the scale here is a bit different. Something's going to have 5 exons, sometimes 6, and so on. But as far as lincRNAs go, it's very highly conserved. And again, positionally, it is always found in the same position. And so it has a single promoter, a single end, and then about 2 kb downstream of that end is the beginning of a gene called CAG2. So what is CAG2? It is a chromatin modifier. So chromatin modifiers are enzymes that are typically using ATP to change chromatin. So for example, to remodel chromatin, move nucleosomes around, and so on. This particular one has been implicated in the position of a specific variant of a histone called histone H43, related to DNA damage in a way that we don't fully understand, and related to neurogenesis. And what's most important from our perspective is that haploinsufficiency. So if you have mutation in one of the 2 copies of CAG2, this leads to epilepsy and autism in humans, right? So for most proteins, it is fine if you have a heterozygous mutation, right? So I'm a carrier of Canavan disease. I have a mutation in one of the 2 alleles of some gene. I don't have any known effect or any known phenotype. But there are some genes, and interestingly, many of them are related to chromatin and chromatin modification, where if you have a heterozygous mutation, so if you lose 50% of the functional protein, that leads to a severe to disease. So in this case, if you have a mutation in one of the two copies of CAG2, with quite high likelihood, you're going to have epilepsy and some intellectual disability. So this happens in humans. Interestingly, in mice, the mice can live without CAG2 quite well. So they have some neurological symptoms. They have some mild phenotypes, but largely they can be quite fine. So we were surprised and excited to see that if we leave CAG2 intact, we don't touch CAG2, but instead we use CRISPR to delete the promoter of chaser, we actually get a severe phenotype. So we cannot get any homozygous knockout mice, and even the number of heterozygous mice that we get is substantially smaller than what we would expect by chance. So we'd expect if we cross chaser heterozygote with a chaser heterozygote, we expect to get 25% homozygotes. We don't get any homozygotes. And then we expect to get about 2/3, a third heterozygotes and wild type. And we instead, most of what we get are wild type. So the mice really don't like to live without chaser. The mice that do survive, they're smaller, and they have a variety of different phenotypes. So, for example, small occlusion where the teeth are growing and the lower teeth are longer than the upper ones. So we have a severe phenotype, and what's causing it, and we can see this in the embryo or in adult tissues, is this massive overproduction of CAG2. So this is wild type mice. They typically don't have a lot of CAG2 protein at embryonic day 30.5. In contrast, mice that are missing just one copy of chaser, heterozygous for chaser, have about 10 times more CAG2 protein. And in adult also, in some cases, it's going to be a little bit more protein. In other cases, it can be a lot more CAG2 protein. And so we can see this in mice. We can also see it in embryonic stem cells, where we can delete the promoter of chaser, and this results in an increase in the expression levels of CAG2. This is on the RNA level here. So it's not just a promoter. We can also delete only the gene body of chaser, and CAG2 is going to go up again by about 60%. For this function, chaser really needs to be produced from the place it is usually produced in the genome. So if we use a different technique and now take just the RNA of chaser and produce it elsewhere in the cell, it doesn't have any effect on CAG2 levels. So it really needs to be produced in the endogenous locus. And more importantly, for any kind of therapeutic applications, we can use an antisense oligonucleotide and repress the levels of chaser. So here we are using different manipulations that are changing the DNA in the cells. Here we're not touching the DNA, we're only knocking down chaser using an antisense oligonucleotide. This is sufficient to upregulate CAG2 expression as well, suggesting that this is really the RNA product that is carrying out this function, but this really needs to be the RNA product or its transcription at the endogenous locus. So how can we be sure that this is really acting in Xist, that is only acting on the chromosome where chaser is produced? So here we could use the fact that in addition to these mice which don't have chaser produced from one of the alleles, we can also have mice that have a mutation in CAG2. So they produce almost no CAG2 protein because they have a mutation here. So if we now take these homozygous mice and cross them with chaser heterozygous mice and then look at the progeny, we're going to have some mice that don't have so we're going to have mice that don't have chaser promoter on one chromosome and don't have CAG2 on the other chromosome. And then we can use allele-specific PCR to ask which of the copies of CAG2 is affected, the copy that is next to the deletion of chaser promoter or the other copy. And here, very clearly in different tissues, we can see that the copy that gets upregulated is always the copy that is next to chaser deletion. The other copy actually gets repressed to some extent. So an increase in the cis allele is accompanied by a decrease in the other allele, and I'll go back to it, but chaser really acts on in cis. And to further prove it, one thing that we could do is we could take these mice that don't produce CG2, and now with CRISPR in these mice, delete the promoter of chaser. So this should be chaser. So slide should have fixed a long time. And here now we can get mice that are completely fine. So they don't have any embryonic lethality, their weights are fine, and they don't have any distinguishable phenotype, suggesting that it's really chaser at the endogenous locus. And that is sort of the chaser phenotype that we saw before is all because of overproduction of CG2, because these mice are producing a lot of CG2 RNA, but they're not producing an overdose of CG2 protein, and then they're fine. And so at least the major phenotype of chaser knockout is because of increase in CG2 levels. And then we spent a lot of time and effort trying to understand why is it so bad to have this overdose of CG2 protein. And to make a long story short, what we're seeing is that there are many cases. So if we look at embryonic fibroblasts that don't have chaser versus wild type fibroblasts, and then we look at what genes are affected, we can see that the typical gene which is affected is a gene like DAPK3 here. So this is DAPK3, here it's exons and introns. And you can see that when chaser is missing, there is less expression of DAPK3. And what makes DAPK3 special is that upstream of it, there is another gene which is highly expressed. This is another gene that's affected, it's called CPT1C. So here as well, we can see that there is a repression in its expression, and upstream of it, there is another gene that is highly abundant. And we can quantify genome-wide, and we can see that so there is preferential repression of genes that have another neighbor on the same strand in close proximity to it. And if this upstream neighbor is more highly expressed, there's going to be even more repression. And in contrast, if we look at this divergent orientation, there is no effect, right? So this is only happening in this particular case of interference. And in contrast, if we look at mice that don't have chaser, but also don't have CG2, again, there is no effect. So specifically, if you have this overproduction, this leads to this kind of interference. And I'm just going to skip this. So this kind of gives rise to our model. So sort of from the beginning of this project, we always suspected that maybe there is some kind of a regulatory feedback loop here. And this really supports it. So the idea here is that in regular conditions, CG2 is being made at quite low amounts. So this is the amount that the cell needs. And part of this CG2 goes back to its own promoter, and together with chaser is repressing it a bit. So we're maintaining it within a certain range. But when chaser is missing, this sort of autoregulatory negative feedback loop is lost. And as a result, we have a lot of production of CG2 protein. This CG2 protein, at least on the allele where chaser is missing, is now unable to repress itself. So it goes into this vicious cycle. It's producing a lot of the protein. If there is another allele of chaser in the cell, that other allele is going to be repressed. So this can help explain why in this setting where we had compound heterozygous mice, one allele was increased and the other one was decreased. So this suggests that in this case, the reason that chaser in all of these different species is always found next to the promoter of CG2 is because it is acting as part of this feedback loop. So it's also a nice example of how we think we learn something new about the protein of CG2 by studying the lincRNA produced next to the promoter. And of course, another reason this is interesting is that there has been a lot of interest in recent years in silencing genes using antisense oligonucleotides, in particular for cases of relatively rare diseases where other therapeutics are not available. And so particularly, there was a lot of interest from this paper from New England Journal of Medicine a few years ago showing that a girl that had a specific genetic change and could be treated, not cured unfortunately, but treated by an antisense oligonucleotide that could target a specific region and repress it. So one thing that we are now sort of pushing forward, and actually there is a company now interested in this and supporting this research, is whether we can use this fact that chaser is repressing CG2 as a potential therapeutic strategy for increasing the levels of CG2 in individuals that are haploinsufficient. And so this is just some first experiment supporting this. So in human cells as well, we can take antisense oligonucleotides that are repressing chaser, and these antisense oligonucleotides are leading to indeed to an increase in the expression level of CG2, and we can see it on the RNA level, and we can see it on the protein level as well. And now we have sort of carrying out additional studies and some other strategies that I won't have time to go into to try and see whether indeed eventually this can be therapeutically relevant, right? Because we're building here on the fact that theindivviduals that are haploinsufficient, so they have a mutation in one copy of CG2, but they have another copy that is just fine. And it's just, there's only 50% of it. So if we reduce chaser and then increase CG2 by about twofold, which is what we typically get, this can be potentially therapeutically irrelevant. And this is a case where other types of gene therapy are much more challenging because CG2 is a very large protein. So it's difficult to build a virus that's going to express it. And then so this also further supports the idea that in this case, so we have a lincRNA that has similar functions in human and mouse. And there is also some evidence that loss of CG2 indeed leads to epilepsy or to motoric phenotypes in human and mouse. And in this case, we also have some unpublished work that there is also evidence that chaser singular copy loss leads to epilepsy and sort of related neurological disorders in human, mouse, and also there is some evidence for that from zebrafish as well. And we think that in mice, this is also associated with this strong growth retardation phenotype mediated by significant transcription interference. Overall, this shows that this particular gene, but this is true for many other chromatin-related proteins, is under tight control, right? So if you have 50% of it, you can get epilepsy and autism. But if you also have an overdose of it, that can also have quite severe consequences. And indeed, we know that many of these genes are very tightly regulated. And so to summarize, there's this famous quote from Donald Rumsfeld that I think just passed away last year, that we can sort of divide the things in life to known knowns, which are the things that we know, known unknowns, that things that we don't know, but we know about them. And then there are sort of unknowns, things that we don't really know, that we're not aware of them. So lincRNAs for until about 10 years ago, they were these kind of unknown unknowns. We're not really aware that they're there. Now we know that they are there. In some cases, they're important. They're challenging to study because there is a lot of diversity. And we so far don't have a very clear division into groups. So for example, we're very excited about chaser and PPT1, but we don't really have a good handle on what other lincRNAs are like chaser, are likely to repress their gene next to them. But there is a lot of things to do in the field, and so we're excited to be working on that. And I'll be happy to take any questions. Okay, so there is one in the chat. This lincRNA doesn't directly interact with the target gene, but instead suppresses it from stealing a transcription machine. Yes. So the question is sort of, can this be an example where there is really a competition between the production of chaser and the production of CG2? So indeed, there are some examples of that in the literature, and there is some evidence that this happens here as well. So if we go back to the model, so we know that if we look in the human genome, so this is chaser and this is CG2, and here there are many enhancers, and these enhancers are activating both chaser and CG2. And indeed, if we look across different tissues, typically chaser and CG2 levels are quite similar to each other. So even though chaser is repressing CG2, there are other processes that are activating both of them similarly. And we also know that if we delete the promoter of chaser, then these enhancers have a higher tendency to come to CG2 promoter. So there really is some competition between them. But on the other hand, we also know that if we delete just the gene body of chaser, then we still have this increase in CG2. So it's not just about this competition. And also, these antisense oligonucleotides we're using, they're reducing the RNA. So they may be reducing transcription here a bit, but they don't have any particular effect on these interactions with the enhancers. So it's not just about competition between the promoters. And we also know that the RNA is highly conserved. So that means that there really is a function for the RNA here. But part of the story in this case is going to be this kind of competition for recruiting the transcriptional machinery. Okay, so Natalia is asking, what are the key skills someone investigating lincRNAs should have? How do we deal with hyperpropagate methods as biologists? Should we start learning R and Python? Yes. And what can we expect in the field of lincRNAs? Okay, this is a good question. Yeah. So as you can see, a lot of the work that we do in my lab is very interdisciplinary. And part of the challenges is that eventually we need to use a large variety of different experimental methods. So that's one challenge, right? Because you could see that we're doing some Western blots to study proteins, and we're doing a lot of sequencing RNA, sequencing accessible chromatin, ChIP-seq. We are also doing some biochemical methods, such as purifying RNA and identifying proteins, interacting with it, and so on. And as well as many other things, right? So that's part of the challenge, that this really requires quite a big arsenal of different methods. Also CRISPR, mouse models, and so on. So not every lab really needs to establish all of this, right? It depends on the kind of questions that are being asked. In terms of combination of experimental work and computation methods, yes. So that's very much something that many labs in biology now are doing. The model in my lab and in many other labs, that the students that are performing the experiments, they're also doing most of the analysis. And they are not students that studied computer science or bioinformatics before. So it takes time, and it's challenging, but eventually most of the analysis is not very complicated. So you don't need to have a degree in computer science to handle it. The learning takes time. And we usually do it in R, but some people in the lab are using Python as well. And so my lab has about 14 people. One of them is really coming from more computer science, like I am, but all the others just studied biology. And then not everybody is analyzing their own data, but many of them are. And there are a lot of so for a lot of the more complicated steps, such as taking the RNA-seq reads and mapping them to the genome and so on, there are already tools out there. So you just need to learn how to run them. And then downstream, eventually it's a combination. So it takes a long time to become proficient and to know how to write the right set of commands in R to give you the answer. But that's definitely something that is possible and doable. And it's never too late to start. Obviously, it's better to gain some of this knowledge early on. So some of you are undergraduates, but it doesn't have to. Like, I think in most cases, this learning is most effective when you have something specific. If you have your own data set, you're much more likely to actually learn and master some of these methods compared to if you just sit at some course and then working on something that is not related. But this is just going to keep getting sort of more complex, right? And so the need for the ability to analyze data and use R and tools from data science. But I think the good news is that you don't necessarily have to have a degree in math or in statistics or in computer science. For some things you do, but for many of these methods, not necessarily. And in terms of sort of RNA biology and lincRNA, so as you saw, there are many open questions, and still we are sort of far from this goal of really identifying families of lincRNAs. But I think a lot of the focus now is mostly on trying to identify the mechanism, right? So there have been already many different phenotypes that have been described, certainly in cell lines, that this lincRNA, you knock it down, cells divide more rapidly, less rapidly, and so on. The challenge is to find mechanisms and to find mechanisms shared by multiple lincRNAs, but also another sort of, from the biomedical point of view, drugs that bind RNA, drugs that affect RNA properties, these antisense oligonucleotides. There is a lot of interest in trying to see how far this can go in terms of therapeutics. Any other questions? Okay.
If there aren't any more questions, I would like to thank you for this wonderful lecture, and it has been a pleasure to have you as one of our lecturers. And I once again want to thank everyone who has attended this lecture, and I hope you will join us for one of our other upcoming events. Goodbye.